Hello, everyone. Um, Good afternoon, everyone. So we are in the last segment of our syllabus. So we have today we have the class. Then the next class we'll finish this course. But already I have set off all your questions, 40 questions, and this time all these questions are from my part, and they are on the slide. They are coming from the slides, no out of syllabus. Very cool. So just go over this. I hope you will do good like the previous exam. So there is an announcement that we have this exam on 14th December at 5:30 to 6:30 here in this auditorium. So please mark that in your calendar. And also I will send you the reminder in the announcement. So we will today talk about this very long topic, but I tried my best to cut it short. Cell death or the apoptosis. So we will, and also in the next lecture I'll be talking about how can you prevent apoptosis by the signaling cascade by neurotrophic effector. So I have highlighted some of these points that I am going to discuss about cell death, general cell death versus apoptosis, that is programmed cell death. And I'll be talking about mostly in the model system C. elegans, which has been analyzed thoroughly about say programmed cell death. Then I'll be talking about the apoptosis, and these are the components of the apoptosis, the enzymes, caspase, and caspase activation. The important organelle that plays an important role in apoptosis, mitochondria, and some of these apoptotic, pro-apoptotic or anti-apoptotic genes belongs to BCL2 family members. Then talking about something about stress, and we'll finally conclude with neurotrophic factor signaling in the next class. So we'll go one by one. So here I have kind of set up this example for difference between cell death and apoptosis. As I mentioned that apoptosis, they are already programmed in the cell. So you have certain sets of signaling cascade which activate and cell undergoes apoptosis, and you have elimination of unwanted cells which are not needed during the development. So and another important point that I forgot to mention, that why you need apoptosis, why you need cell death. So when you have during the development throughout our class, starting from the patterning to differentiation, synaptogenesis, we talk a lot about production of the neurons. So always you have production of a lot of neurons, but not all the neurons remains at the final stage. Those are died, they are eliminated during the development. That we call as apoptosis. That's why you need the apoptosis sometimes during the development to basically select the specific group of cells at the final stage of the development. So there you have essential role of apoptosis. Sometimes the damaged cells are removed by the apoptosis programs. Okay, so we'll talk about more of this about the developmental program cell death, apoptosis. So these are the general cell death mechanism when you have DNA damage, or you have hypoxia, or osmotic stress, or thermal stress, or exposure to, of this organism or cell to the noxious or toxic substances. So these are not programmed cell death. Okay, so these are called simply cell dies, and you have elimination of stress cells. If those cells will remain in the organism, that will create a mess. Okay, so we'll basically deal with this the signaling, highly orchestrated signaling cascade, the developmental apoptosis during the development. So, and this can be two different types. These are genetically programmed. So here everything is genetically programmed. Suppose cell undergoes some stress, or there is some kind of aggregation of some proteins. So the cell actually initiate certain programming, certain signaling cascade, which are genetically programmed. So you have removal of these cells. So these are actually target independent, and sometimes they are target dependent. Suppose you have many presynaptic neuron and you don't have postsynaptic, like dendrites. So then, those presynaptic neurons, some of the presynaptic neurons will be removed, and these are, target, dependent. So you do not have the target, the postsynapse. That's why you have removal of this presynapse, the presynaptic cells. So those are target independent. And sometimes the axon projected to the wrong target, the axons will be removed. And as I mentioned, the excess of cells in the presynaptic pool, the excess cell in the presynapse, the presynaptic neuron will be removed because you don't have same amount of postsynaptic dendrites. So those are target dependent cell death. And mostly the genetically programmed or developmental programmed cell death are target independent. So we'll deal with these two things. So this is the done. So then we'll talk about some model system where it has been discussed about program cell death during the development. Okay. So here, before going detail about that, the program cell death in some model system like C. elegans, I want to highlight, the neuron dies during the development. And here there are some examples. Here, this is example for the cheek spinal motor neuron or cheek peripheral autonomic ganglia, or during human spinal cord development or brainstem nuclei. So here, you can see that during the cheek spinal cord motor neuron, you can see that this is up. So this is before hatching. These are the days where you have an incubation period, development of this cheek embryo during in the egg. So you have 5 days, 10 days of incubation, 15 days of incubation, or 18 days of incubation. You can see that 5 days of incubation, you see large number of cells, neuronal cells. And it goes down when the development progress. And even after hatching the embryo, you can see that you will, less number of neurons. So you can see that, how this process is highly regulated. So a lot of neurons dies during the development, and that is programmed. That is already programmed. So these are called as programs cell death during the development. So here some of these examples, you can see that like during the development, a lot of neuron dies, and these are not random. These are programmed cell death. Okay. So this study of the programmed cell death was initiated by Rita, Levy, and Victor sometimes around 19th century, and they hypothesized that the cell number control, the number of cells in the presynapse or in the postsynapse controlled by the target selection. So suppose you have a large number of presynaptic cells, you do not have that much amount of postsynaptic dendrite, you will have death of the presynaptic excess presynaptic cells. So that means your survival of the neuron here depends on the target. You do not have that much target. That's why. So they, here this is the example of cheek embryo development by Victor. They have identified that the control of neuron number depends on the target. So here you can see that this is cheek embryo, and here you have this limb board. So when they remove the limb board, you can see that you have less number of you have fewer neurons here. You can see that when they remove this limb board, you can see that you have fewer number of neurons. But when they add the limb board to this cheek embryo, you can see that you have more number of neurons. So it depends on the target. If you remove the target, you have production of less neurons. So they are target dependent, and this work is done by Hamburger in 1934. And the same thing is also shown here. Like this is again in the cheek embryo. This is the dorsal root ganglion. This is the left side and the right side. So when they remove the leg during the development of the cheek embryo, when they remove the leg, basically the dorsal root ganglion project to the leg. When they remove the leg, here they remove this right leg. You can see that you have less number of neuron in the dorsal root ganglion. So when you remove the target, the leg, you have less production of neuron. Similarly, here when they have done same experiment with the other part of this body, like cheek embryo. Here, this is the right side of the branchial ganglion. This is the left side of the branchial ganglion. When they remove this wing in the right side, you can see that cheek embryo wings. So this you can see that you have less number of neurons. That means the target is here in the wing. So and the neurons are controlled. The number of neurons are controlled by the target selection. Okay. So this is all about how the neurons are controlled by the targets. The number of neurons, the production of the neurons are controlled by its target, like either it is leg or is by the wings removal. Okay. So the two classical experiment. So here again, same thing kind of, I'm showing that how, the neurons, number of neurons is controlled by the target. So here, this is the cheek embryo spinal cord. Here you have more number of motor neurons which are overproduced when both limbs are innervated. When you have both limbs in the cheek embryo, you can see that you have large number of neurons. So again, the same thing that I'm showing that in the experimentally, what they have done is they have surgically removed this limb board, and you can see that you have missing when you have missing of this limb. You can see that you have less number of neurons, and this can be rescued or suppressed, this phenotype, when you replace or you kind of transplant this limb bud. Here you can see that you have production of neurons, rescue of this phenotype. That means the number of neuron number or number of neurons in this spinal cord is controlled by the target. Okay, so we talk about like cell death, normal cell death, then you talk about the programmed cell death, then how this neuron number or the cell number is controlled by the target. So we'll talk about some more about the neurotrophic hypothesis and what are these. So here, as I mentioned in the earlier section of my talk today, that you have a lot of production of this neuron during the development, and those are getting eliminated when the latter stage of the development. And this happens because of the competition between the neuron to find the proper its neurotrophic factor. So here you can see that we have large number of input neurons, and here this is the target. These are the target neurons. So if they get the neurotrophic factor, here they are getting you can see that you have 1, 2, 3, 4, 5, 6, 7 neurons. Here you have only the 4 neurons are survived because these neurons get that they are proper neurotrophic factor. That's why you have survival of these neurons. These neurons are eliminated because they cannot compete to get this neurotrophic factor. So that means the neurons number is maintained by its neurotrophic factor. There is a competition among the neuron to get the neurotrophic factor. Okay, these neurons which get insufficient neurotrophic factor, they die. They undergo apoptosis. Okay, and they are tightly regulated by programmed cell death or programmed cell suicide. Okay, so we'll talk about this. So these are some of the important points of the neurotrophic hypothesis. So neurons are initially overproduced during the development, during the early stage of the development, and the neuron project to their target and encounter target derived neurotrophic factors. So they project to the targets, they compete among themselves, among themselves. So the neurons which get the target neurotrophic factor, they survive, they remain in the system. Other neurons which could not compete to the neurotrophic factor, they eliminated. Okay, so like here we'll be talking about apoptosis, more details, and sometimes what happens is neurons which receives less neurotrophic factor, then they have a greater probability of undergoing apoptosis. So go over this slide. These are all these points that I mentioned during, these are the summary things that I talked about. So we'll talk about more about programmed cell death in one of the favorite model system that people have used, and they got Nobel Prize for this by working in C. elegans, finding the mechanism of programmed cell death during the in C. elegans. So, and these are the people who share Nobel Prize Physiology in Medicine in 2002. Sidney Brenner, then John and Bob. So they work on the programmed cell death in C. elegans, and this is the statement that I made in the initial some of my initial talk, that progress in science depends on the new technique, new discovery, and new idea, probably in that order. So you develop new techniques and develop discover certain new things and new idea. You have generation of a new idea, and that is how the science works according to Sidney Brenner. Okay, and yeah, so here they talked about how the cell number is regulated in C. elegans. So here they took the these are the different progenitor cells in C. elegans, and they develop different structure in the larval C. elegans larvae. So C. elegans larvae there are 4 stages of the C. elegans development. Larval stage. One is the L1, first instar, then second instar, third instar, fourth instar. So L1, L2, L3, and L4. So, and takes around 40 hours to get L4 larvae, L4 larvae of the C. elegans. So here you can see that these are the different progenitor cells, and they divides to give many number of cells. At 10 hours, mostly in the L1 stage, you can see that you have more production of these cells. But as the time progress, some of the cells selectively die, which are also, you can see that some of the cells selectively die, and this happens because you have initiation of this developmental program, program cell death. So we'll talk more about this in the coming slides. What are these genes required for the program cell death in C. elegans? And there are homologs of the gene present in humans, which also required for the program cell death. So here, this is a pathway for the program cell death in C. elegans. So these are the protein, these are the genes like SED9 genes or SED9 genes, EGL genes, SED3 genes. So these are the genes. They get activated during when the there is program cell death in C. elegans. So what happens is the SED9 expression. When you have more expression of SED9, what it does is it inhibits the expression of EGL1. So SED9 inhibits the expression of EGL1. Then EGL1, if it inhibits the expression of EGL1, then you do not have production of more EGL1, or you have dysfunction of EGL1. EGL1 cannot activate SED4, or SED4 cannot activate SED3. Then you do not have apoptosis. So that is how this is the pathway. So here this is the typical pathway. So when you do mutation in the SED9, what happens is you have increases, you have more cell death, because when you mutate this, you have always overproduction of these proteins. You have more expression of EGL1 because SED9 inhibits the expression of EGL1. When you have mutation of this, you do not have enough of this to block this one. So you have more production of this one, you have more expression of SED3, then you have more apoptosis. So these are the genes which I mentioned that SED9 mutation increases the cell death. Okay, so here this is wild type C. elegans. You can see that you have many number of cells are produced here. But in the SED9 mutation, you can see how many cells are produced. So here, like the divide again, divide. Here you can see. So the SED9 actually, SED9 mutation, SED9 regulates the cell death. Okay, so when you have mutation of SED9, you have more expression of EGL1, SED4, and SED3. It increases the apoptosis, program cell death or apoptosis. So remember, these are the genes which are required for the apoptosis. I might ask questions, in this slide. So just remember this EGL1 or SED9 or SED3 or SED9. Okay. So we'll talk about more about apoptosis and this enzyme caspases, the main enzyme that required for the apoptosis. So I'll before going details about the detailed process of apoptosis, I want to highlight the important enzyme, the caspases, and that actually regulates apoptosis process. So this is the enzyme caspases is a 16 aspartate protease. Okay, so it cleaves after it cleaves the substrate after each aspartate residue. Okay, so here it can also act on the DNA. It can also, you can see that it can cleave the DNA, or it can disassemble the cell structure or other proteins, regulatory proteins, or other important proteins. Like here in this protein, you have the catalytic domain, you have the regulatory domain, and this is the aspartate residue which act on by the caspase enzyme. You can see that you have cleavage of these proteins. So these are the caspase cleavage sites. So it has been seen that it acts around 1300 substrate protein to kind of cleave these these proteins to different fragments. So here you can see. So this is the mechanism how to work on like during this during these processes. So here the caspases can also process the zymogen. These are the pre-enzymes when you have cleavage of these the segments, you can have the production of active caspases or active enzymes. Okay, so these are the zymogen or the pre-caspases. It will become processed and become active caspases. So that is how the importance of caspases for yes.
A question about the cleavage. Is it that the caspases cleave other proteins and then those proteins start cleaving others? Or is it that it just cleaves and that protein no longer works?
Yeah, it cleaves the protein. Yeah, it cleaves the protein. So here this is showing that how caspase is processed actually. So here the active caspases, it is these are the zymogen, the inactive caspases. So how it become active caspases. But caspases act on other protein to kind of cleave and degrades. So here this is the example. Okay. Next, we'll talk about some more about the programmed cell death. So however, the caspase knockout mice are viable. So here this is the wild type mouse having the caspase intact, and this is the knockout mouse. The caspase is not intact. So you can see that they are viable, but if they have larger head, because caspases actually induce apoptosis. If you do not have apoptosis, you have more production of neuron, and you have bigger brain, so which is not fitting to this in mice without functional caspases, neurons do not die. They do not die because you require caspase activation for the regulation of the cell number. If you do not have the caspases, what happens is the neuron grows like anything, and you have fetal brain. It's too large and it's not fitting to the skull. So that's why you have larger head. Okay, so caspase knockout mice, they are viable, but they have larger head. They have more neurons. Okay. That is what is showing. So here these are the important steps in the programmed cell death, and mitochondria plays an important role in programmed cell death. We'll be talking about these things. So the important organelle that is kind of interest to me and also other people like working on the mitochondria. So the mitochondria plays an important role during the apoptosis. What happens is mitochondria during apoptosis, what happens is mitochondria, you have two membrane, the mitochondria, you have inner membrane, you have outer membrane. So you have permeabilization of the outer membrane during apoptosis. When you have generation of these signals, what it does is there is a permeabilization happens in the outer membrane, and you have activation of these proteins, that BCL2 family proteins. So BCL2 family proteins belongs to they are also pro-apoptotic or anti-apoptotic. So the pro-apoptotic proteins are BAX protein, and anti-apoptotic protein are BCL2 and BCLXL. Anti-apoptotic means it inhibits apoptosis. Pro-apoptotic means it initiates apoptosis. It induces apoptosis. Okay. So what happens is you have the activation of this protein, pro-apoptotic protein, that is BAX protein and BAC protein, and they undergo conformational changes, and they recruited to the membrane, and they form the pore. They helps in permeabilization of the membrane, outer membrane. When you have permeabilization of outer membrane, you have release of cytochrome C, and mitochondria has the reservoir for cytochrome C. So you have release of cytochrome C to the cytoplasm. Now in the cytoplasm, it interacts with a protein called as APAF1, and this is a apoptotic protease activating factor 1, and it forms with procaspases. It forms apoptosome complex. Okay. So you have permeabilization of the outer membrane, you have formation of pores by the BCL2 family members like BAX and BAC. You have release of cytochrome C from the pores to the cytoplasm, and it associates with APF1 and forming this apoptosome complex. And that complex basically activates the caspases, downstream caspases like caspase 9, caspase 3, and it acts on these targets to kind of degrade the target. So we'll talk about this in the diagrammatic representation. So here I'm just kind of showing. Okay. So what happens here is in the mitochondria, you have permeabilization of this outer membrane, you have release of cytochrome C. You can see that the release of cytochrome C, these are the BCL2 family members, which helps in the formulation of this membrane, and you have release of cytochrome C. You have association with the APF1, and it forms this apoptosis complex and activates caspases. And caspases does a lot of things. It can damage the DNA. It can damage the cell membrane, cytoskeletal proteins. You have degradation of these cells, or death of cells, which is engulfed by the macrophages and microglia. So this is how the mitochondria plays an important role during apoptosis. So, and there are certain inhibitors or activators for the apoptosis. So basically, these are the inhibitor for caspases, which induces apoptosis. So these are the inhibitor of apoptosis are these are the IAP proteins. Okay. So and there are activators of apoptosis. So you have this protein called as diabelloprotein, which can also inhibit these inhibitors, so that you have activation of caspases, and you have production of this degradation of the certain the membrane, the structure, the cytoskeleton, all these things. So there are inhibitors and activators. Okay, this kind of highlights that and human neurodegenerative disease called as spinal muscular atrophy is linked to the loss of functional mutation of IAP family members. So these inhibitors of apoptosis basically. So if you have mutation of this IAP protein, what you can in this basically in the spinal muscular atrophy, it has been seen that there are certain proteins belongs to the IAP family members. You have inhibition of this. You have what happens is the caspases remain active, and it can kill the neurons. Okay, so these are the difference between C. elegans and mice programmed cell death. So in the C. elegans, you have SED9. In the mice or in the mammals, you have BCL2. You have activation of a caspase 9 and activation of a caspase 3, which we do not have this in the C. elegans. The SED3 plays an important role directly involved in apoptosis, but in the mammal, you have caspase 9 and caspase 3. Okay, and these are the inhibitors for the caspase 9, the IAP, and the IAP is again inhibited by the diabello. So these are the inhibitor for the IAPs. So it's diagrammatically represented. Okay, so BCL2 family members are anti-apoptotic, and also they are also belongs to pro-apoptotic members. So, okay, so I am not able to pronounce it. So so we'll talk more about the BCL2 family members of apoptotic regulators. So here the process showing that how these BCL2 family members regulates apoptosis. Okay, so here these are the back and backs. They are the pro-apoptotic and they are anti-apoptotic. They inhibits apoptosis. Okay, so when you have activation of these, what happens is the release of cytochrome from the mitochondria. Same thing kind of repeating, and it associates with APF1, forming the apoptosis complex. Then you have activation of caspases, and caspases activation of caspase 9. The caspase 9 activate the caspase 3, 6, 7, which can degrade the cell membrane or the different structure of the cells. Okay, and these are the inhibitors of apoptosis. These are the anti-apoptotic like BCL2 or BCL-XL. So we'll talk more about these things. So here this is the pro-apoptotic and anti-apoptotic. Okay, so we'll talk more about this thing in the coming slides. Then we will talk about We'll talk about how apoptosis is controlled by neurotrophic stimuli. So neurotrophic factor basically inhibits apoptosis. That is going to talk about. And what are these neurotrophic factor? So here at the mitochondria, you have production of this BCL2, activation of the BCL2 family member proteins. These are kind of pro-apoptotic proteins. So here what happens is you have there is certain set of signaling cascade which regulate the transcription of this BCL2 family member protein, which are pro-apoptotic. Okay, you can see that they are pro-apoptotic, and when they are produced, the release from the cytoplasm. So we'll talk more about the mechanism, how these are produced. The release in the cytoplasm, and they also some of this stage in the cytoplasm, they also they bind to this mitochondria, forming the pores, and you have release of cytochrome C, and you have downstream process. And similarly, this those processes that here showing the apoptosis can also be inhibited by the neurotrophic factor, the signaling factor, which are anti-apoptotic. So we'll be talking about these things. Some of these anti-apoptotic pathway that can regulate then inhibit the apoptosis. Okay, so here you can see that these are the anti-apoptotic, and it recruited to the mitochondria. They inhibits this this pathway, the apoptosis pathway. Okay, so they belongs to this family member, the AKT protein kinase. Basically, these are protein kinase B. They are anti-apoptotic or ERK subfamily members. Belongs to extracellular signal regulated kinase. Okay, so they actually can inhibit the apoptosis process. So we'll be talking about these things, and they are synthesized. And they're transcribed and translated from the proteins, and they're translocated to the mitochondria to inhibit the apoptosis. So we'll talk about these things. And here, kind of showing the regulation of this neurotrophic factor signaling, how it can actually inhibit apoptosis. So here, this is the neurotrophic factor receptor. So we'll talk about this more of this receptor in the next class. But for the timing, just feel that this is the receptor, and you have binding of this neurotrophic ligands. When you have binding of the ligands, you have activation of the ERK pathway, which is extracellular signal regulated kinase pathway, and this ERK can activate the AKT, or directly this receptor can also activate PI3K kinase pathway. It can activate AKT or protein kinase B. So when you have activation of this protein kinase B, you have phosphorylation of the protein called as bad protein. When you have phosphorylation of this protein, it remains sequestered in the cytoplasm. Okay, so it cannot recruit it. This is again pro-apoptotic proteins which can induce apoptosis. So when you have phosphorylation of this, because of activation of this pathway, it remains in the cytoplasm, and the cell doesn't undergo apoptosis. Okay, this is how you have the neurotrophic factor signaling can inhibit the apoptosis by activating this pathway. Okay, by phosphorylating the bad proteins. But when you withdraw this neurotrophic factor signal, the factor receptor or the signaling, what you have is you have dephosphorylation of this bad. Again, when you have dephosphorylation of the bad, it can translocate it to the mitochondria, again induce apoptosis. Okay, so that is what showing that how neurotrophic factor signaling regulates apoptosis process. So, and these are both transcriptionally and translationally regulated. Okay, so those pathways transcriptionally and translationally regulated. So where here you have transcription, there are certain transcription factor required for the transcription of pro-apoptotic genes or anti-apoptotic genes. So these are translationally regulated. Then after transcription, so these are transcriptionally regulated. Then after transcription, you have translation of these proteins. So they are both kind of transcriptionally and translationally regulated. So increased synthesis of pro-apoptotic regulator promotes apoptosis. So when you have more synthesis of pro-apoptotic genes or proteins, and mostly the proteins, then you have more production of more death of the cells. But when you have synthesis of anti-apoptotic regulators, mostly the activation of the neurotrophic factor signaling, you have less apoptosis. Okay, when you have more products, synthesis of pro-apoptotic genes or proteins, you have more apoptosis. But this can be suppressed by activation of the neurotrophic factor signaling. That is one to kind of highlight it. So they are both transcriptionally and translationally regulated. So, and here when the cell is undergoing apoptosis and you want to stop apoptosis, if you can induce the cells with neurotrophic factor signaling, so there will have a competition between this pro-apoptotic and anti-apoptotic pathway. So if you can if you can induce more anti-apoptotic pathway, it can compete to the apoptotic pathway, then you have survival of the cells. So that is what you can induce the positive feedback, so that you can inhibit the apoptosis. So these are by all these are regulated by transcriptionally and translationally. Okay, so here showing that how these are regulated by the transcription factor. So here I'm showing one of these pro-apoptotic genes, that is BAX genes. or back genes. So here I'm showing with back genes. So what happens is when you have activation of backs, so how the backs is activated. So you have generation of this pathway called as JNK pathway, which is a June N-terminal kinase pathway. So the JNK pathway, induces a transcription factor called a June transcription factor. That's why I'm saying that they are both transcriptionally and translationally regulated. So here, this is the transcriptional regulation. So we have activation of a JNK pathway, and we have formation of a June transcription factor that binds to the DNA and that transcribe the DNA and forms this mRNA, which translated to form this Bax protein. Okay, so transcribe this DNA, which can form the mRNA for Bax, then Bax, translated to from the Bax pro-apoptotic protein, which again recruited to the mitochondria, forming helps in permeabilization of the mitochondrial membrane, so that you have release of cytochrome and induction of apoptosis. Similarly, so you can have also, we can have also the other transcription factor, which can also induce the expression of anti-apoptotic proteins, which can inhibit these processes. So again, they are also transcriptionally, translationally regulated. Okay, we'll talk some of these, some of these processes, like how these are being regulated, tightly regulated in the cells. Okay, so you have activation of a neurotrophic factor, which can also block this apoptosis. Or when you remove this, withdraw this neurotrophic factor, you have activation of this pathway, the JNK pathway, and production of this Bax protein, which can promote apoptosis. It depends on this, what kind of thing you want going to try. If you remove this neurotrophic factor, or withdraw the neurotrophic factor, you have production of this, pathway. You have, formation of JNK, and activation of a JNK pathway, which can produce June transcription factor, and it can initiate the transcription of Bax mRNA. Then Bax mRNA can translate it to Bax protein. It can insert to the mitochondrial membrane, which helps in the forming the pore or release of cytochrome C from this mitochondria and from activation of these caspases. Okay, so and these are some more example of the protein like beam protein, like how another pro-apoptotic protein, beam protein, can also regulate apoptosis. So they have the different, they use different sets of transcription factor, like it can be June, FOXO, or MAOB, all these things. So then, what is the mechanism of production of this anti-apoptotic transcription factor? So here you have the receptor called as a neurotrophic receptor, which binds to the neurotropins. And when you have binding of neurotropins, you have activation of a RAS and RAP signaling pathway, which can signal ERK. ERK translocated to the nucleus, and it binds to Krebs transcription factor, activate Krebs transcription factor, and Krebs transcription factor binds to DNA and transcribe the anti-apoptotic BCL2. And when you have production of BCL2, it can recruit it to the mitochondrial membrane. It can inhibits the release of cytochrome C from the mitochondria. Then you have you can actually stop this apoptosis process. So this so that's why the importance of neurotrophic factor signaling is it stops apoptosis. Okay, so these are all tightly regulated. So here showing that they are first transcribed, then translated, forming these BCL2 proteins and recruited to the mitochondrial membrane. And this is this happens because of the activation or binding of the Krebs transcription factor. Okay, so you have this ERK signaling that initiates kind of does these processes. So ERK, what it does is when you have binding of the neurotrophic factor to the neurotrophic factor receptor, then you have RAS, initiation of RAS signaling. The RAS activate the ERK, and ERK does a lot of things. It activates the CAM kinase, which can phosphorylate protein kinase A. Protein kinase A activates cyclic AMP. Then cyclic AMP, because of activation of cyclic AMP, it recruited the Krebs transcription factor to the DNA. Then it can produce BCL2 mRNA, which can translate it to form BCL2 proteins, which is anti-apoptotic proteins. Okay. So this is the complete all the processes showing that some of these transcription factor, Krebs transcription factor for the neurotrophic factor signaling, June and FOXO for apoptosis signaling, and these are the receptor, these are the kinases actually. These are ERK kinases for the anti-apoptotic pathway, or AKT, PKB, or JNK for apoptosis pathway. Okay, these are the enzymes plays an important role during this apoptosis or programmed cell death. So we'll talk more about some of these neurotrophic factor, which kind of inhibits apoptosis, and this is really exciting and important for the development, kind of to control the cell number. So in the body you have always regulation of the cells by because of the apoptosis and also because of inhibition of apoptosis, and they're highly programmed. So we'll talk more about this neurotrophic factor, how the neurotrophic factor, fast neurotrophic factor, the NGF, the nerve growth factor, is discovered. So these are the person who discovered this neurotrophic factor, Rita Levy and Stanley Cohen. So they share Nobel Prize in Physiology and Medicine in 1986, and they identified the fast neurotrophic factor, the nerve growth factor. Okay, so it promotes neurogenesis, or it promotes kind of maintenance of the growth cone or neurites, and also it inhibits apoptosis. So a lot of experiments they have done. So here what they have done is here when you have a loss of this limb board, you have a loss of this neurons in the spinal cords. So that was when they kind of transplanted the limb board, you can see that you have decreased cell death and you have production of more neurons. So when you remove this, you have more cell death, and this is, we talked about the target selection. Okay, and when you have this put this limb board, they have more neurons because the limb board secretes some kind of factor which is required for the maintenance of the neuron, and this is what they have identified is this NGF, nerve growth factor, in the limb board. Okay, so here so another example showing that how this secreted factor plays an important role in the growth of the neuron. So here this is the spinal cord, and you have the sympathetic neurons, you have the sciatic neurons. So when experimentally it has been seen that when they place this sarcoma close to these neurons, you have more formation of this connection, you have more formation of you have more growth of this neuron, and that happens because the sarcoma secretes some chemical substances which promotes this growth of the neuron. So these are those are identified as the nerve growth factor promotes this growth of the neuron. So we'll talk more about this. What are these factors? Another example, it has been seen that when they place this tumor which is more vascularized with many blood vessels, okay, in the developing embryo, the chick embryo, they have found that this tumor secretes some factor which helps in growth of this sympathetic neurons. So you have more growth of these neurons, and those factor are called as the they promotes the nerve growth. That's why they call it nerve growth factor. Okay, so we'll talk more about this, and this factor is basically this structure. The tumor is secreting basically some factor, a maintenance factor, which is actually helping in the growth of these neurons. So we'll talk more about this. What are the neurotrophic factor? What are the factors that is secreted from this, either from the limb blood or limb board or from the sarcoma? Okay, so here they have done two experiments where they have taken this control, and here they have not added any sarcoma, and these are the kind of some sympathetic nerves. Okay, so what they have done is they have cultured this with sarcoma, directly with sarcoma, and or with sarcoma with sarcoma conditional media. You can also culture your neuron in conditional media. Conditional media contains important secreted factor which helps in growth of the neurons. So when this culture these neurons with sarcoma conditional media, you can see that you have more growth in the neurons. That means those secreted chemicals promote neurite growth. Or when they co-culture this sympathetic ganglion with sarcoma, directly with sarcoma. So here they culture directly with sarcoma. Here they culture this sympathetic ganglion with conditional media, the sarcoma conditional media. So in both the cases, you can see that you have more neurite growth in these, ganglions, suggesting that, the nerve growth factor plays an important role for the growth of the neurites. Okay, so here, then what is that secreted, the next question is, what is that secreted, substances or secreted molecules from this sarcoma? So is it a protein or is it a, kind of nucleic acids? The next question is, and they have done few experiments where they found that it is not the nucleic acid, it is the secreted proteins. So here, what they have done is they have taken this, sarcoma, and they have added this sarcoma conditional media with sympathetic ganglion. You can see that you have formation of this, you have more neurite growth. Then they have added in the conditional media, the sarcoma conditional media, with in the sympathetic ganglion with snake venom. The snake venom actually is contains has an enzyme called as phosphodiesterase, which can cleave the nucleic acid. So if it is a nucleic acid which is inducing neurite growth, then you should not see any neurite growth. But you are seeing neurite growth because because it's not a nucleic acid, it is a secreted protein. Okay, that is why you do not see any problem even if you put the snake venom, because snake venom contains phosphodiesterase enzyme which can cleave the nucleic acid. So you have neurite growth because then this suggests that basically it is a secreted protein which is a nerve growth factor which is regulating the neurite growth. In another situation, you can see that when they added only snake venom to the control, even you can see more neurite growth. That means the snake venom contains some secreted chemicals, and which I later want to will show you that that is promoting even more neurite growth in the absence of sarcoma conditional media. What are those factors? So snake venom does not remove neurite growth; it promotes neurite growth because snake venom contains some substances which can also induce neurite growth in the absence of conditional media for the sarcoma. Okay. So it promotes even more than the conditional media. Okay, so here some more example showing that what is that factor, and finally they identified that this is a nerve growth factor. So here this is the sympathetic chain of neurons of 12-day-old mouse. So this is here the control, and this is the experiment. Here, when they treated this with nerve growth factor or the nerve growth factor, or with these are from the snake venom. When they treated this with snake venom, and which contains nerve growth factor, you can see that more growth of the ganglion. You can see here there is more growth of this in the experiment than the control. And the basically the snake venom is derived from the snake venom gland. Okay, and these are evolutionary structure from realizing that the venom glands evolved from the salivary glands. So the venom gland of snake is basically evolved from the salivary gland, and salivary gland contains the nerve growth factor. That's why in the snake venom you have nerve growth factor. That's why you have more production of the sympathetic ganglion. Okay, so that is how the nerve growth factor is identified. You can see that in the 20 days old mouse culture, 20 days old mouse or 4-month-old mouse, when they added this this nerve growth factor, you have seen more production of the sympathetic ganglion. And when they have used an antibody for nerve growth factor, like antibody for NGF, so here in the experiment you can see that they block this receptor for nerve growth factor. You can see that there is less production of the sympathetic ganglion compared to the control. Control, you have this intact signaling processes for the NGF and nerve growth factor signaling. That's why more production of a neuron. But when they cultured these neurons with antibody for the nerve growth factor, you can see that you have reduction in the formation of neuron here in the after 4 months or in the 20 days. So that means the nerve growth factor is important for neurite growth signaling. Okay, so next class we'll talk about some more of this, but I just want to highlight what are those neurotropins, what are these nerve growth factors. So there are various neurotropins. One of them is nerve growth factor. Then you have the brain derived neurotrophic factor, you have neurotropin 3 and neurotropin 4. So these are the neurotropins are not, basically plays an important role in the neurite growth. Okay, so we'll talk, and this is how it looks like, and they form the dimer, the neurotropins. Okay, so and these are the receptor for those neurotropins, or no, you have for NGF, NGF nerve growth factor, you have the receptor like TRKS or P75 NTR. So we'll talk more about this. So we'll talk more about this receptor. So here showing that these are the neurotropins like BDNF or NT3 or the NGF. So these are the they are synthesized as a precursor. Then after catalytic processing, you have the matured molecules like brain-derived neurotrophic factor or NT3 or NT4 or NGF, and these are the receptor for these neurotropins. So you have TRKB for the BDNF, you have TRKC for the NT3 or TRKA for the NGF. So we'll talk more about this factor in the next class. And just remember that the activation of neurotrophic factor signaling, it kind of inhibits apoptosis, and activation of this this BCL2 family proteins or pro-apoptotic pathway promotes apoptosis. So this is all about apoptosis and neurotrophic factor signaling. So we have one more class kind of for the neurotropins. So we'll talk more about this in the next class. So if you have any questions, let me know. Thank you.